Welcome everybody to my presentation with the title "We Will Soon Find Martian Life." As you may have guessed correctly, the title slide is indeed a fake. We are not yet currently walking on Mars as human beings, but we are trying really hard to find any evidence that life still exists on Mars or that life has existed at one point. And this is also about what my presentation is about. Here you see the table of contents. It gives you a rough outline about what I will be talking about in the next few minutes. So let's just start with my chosen research paper. My paper has the title Deep UV Raman Spectroscopy of Carbonaceous Precambrian Microfossils, Insights into the Search for Past Life on Mars. Quite a mouthful, but basically it's a study on deep UV Raman spectroscopy, which can be seen as a back support for the ongoing Perseverance rover mission on Mars. The Perseverance rover has an instrument called Sherlock, and Sherlock uses deep UV Raman spectroscope to analyze and characterize soil samples from the Martian surface. But why is this anyways important? Well, the question of if there life exists at some place other than Earth in the universe and how the life in the future of Earth may look like are major research questions in astrobiology. We know that Mars is located at the edge of the habitable zone of our solar system, and that the only other planet in this zone is Earth. Therefore, our best guess for any planet hosting life is Mars at the moment. We also know that there are dry riverbeds and craters on the surface of Mars, and have seen that there are indicators of water being present in the past. We know that there are nutrients and that Mars had a denser atmosphere around 3.5 billion years ago, which was protected by a magnetic field. And this all leads us to the possible existence of life at one point in the Martian history, and also to the fact that microorganisms are able to adapt to extreme conditions and could be still living on Mars. As a paper by Hornick goes more into depth of this, called Astrobiological Aspects of Mars and Human Presence, it states that the knowledge that life existed would tell us that Mars might be able to support life again in the future, or already supports it. Therefore, knowing any issues which could concern human health and well-being as well must be investigated before humans can walk Mars. Furthermore, the import of internal and external microorganisms which accompany any human mission or are brought purposely to Mars as a part of a bioregenerative life support system need consideration in order to protect Mars in its current condition and in its future conditions. What do we have then so far? So far there was no microscopic evidence for life on Mars. Therefore, there is no definitive proof that Martian life exists. But we have evidence that there might be some life, as seen in a paper of McKay called The Search for Life on Mars. It said that we have evidence in meteorites from the late heavy bombardment that liquid was once present. And as we know, where there is liquid, there also might be life. We also know that organisms can survive in space or simulated Mars-like environments. Therefore, our current strategy for life detection on Mars is the fossil record of Precambrian life on Earth, consisting of microfossils in stromatolites and microbially influenced sedimentary structures. On the slides you will see a stromatolite, which is basically cut, and you can see the different sedimentary layers with the microfossils embedded in between. So this is for now how we can study it with this fossil record, but this study looks into how we can microscopically find any evidence using deep UV Raman spectroscopy. Let's go to the mission. Currently there is the Mars 2020 Perseverance rover active on Mars. Its main mission is to seek signs of ancient life. It launched on July 30, 2020 and was launched by NASA. And it landed February 2021 in Jezero crater on Mars. On the slide you can see a nice simulated picture of the rover in its environment. It has a device called Sherlock, which stands for Scanning Habitable Environment for Raman and Luminescence for Organics and Chemicals. Sherlock is a robotic arm-mounted instrument which can be used for high sensitivity detection, characterization, and spatial resolve correlation of trace organics, biosignatures, and minerals. Its main missions is to scan the Martian surface and near subsurface in boreholes and investigate samples from rock outcrops. It then takes macro and microphotographic images using laser spectroscopy. For that, it has a 50 to 100 micrometer laser spot and has a micro mapping mode which uses 40 laser pulses, which gives us a high density maps of 400 Raman spectra. Therefore, it is used to scientifically understand the current Martian condition and the history of Mars. On this slide, you can see Sherlock, which is the ultraviolet spectrometer, and it's accompanied by Watson, as usual, which is the camera taking the pictures. Again, the rover landed in Jezero crater, which is a crater on the western edge of the Isidias Planitia flat plain, north of the equator of Mars. It is 45 kilometers wide, and scientists think that it was once filled with water. To be exact, for 3.5 billion years ago, river channels spilled over the crater wall and created a lake, and the water carried clay minerals from the surrounding area into the crater lake. Therefore, microbial life could have lived in Jezero during one or more of these wet times. And if this is true, then we can find signs of the remains in the lakebed or in shoreline sediments. Therefore, rock and soil samples from the crater might have preserved these signs and could answer the question if life existed on Mars. Also in the picture you can see the landing site inside the crater. But now, why do this all with Raman and not with infrared or visible light methods? Raman spectroscopy is thought of to be a non-contact and non-destructive technique, meaning it does not alter the matter that it analyzes. It can also detect and characterize organic compounds and uses for that a deep UV wavelength, which is smaller than 250 nanometers. But why is it better than infrared or visible light methods? In a paper written by Abby called Deep UV Raman Spectroscopy for Planetary Exploration, the search for in situ organics, both methods are basically compared. And the takeout from this paper is that the mineral transparency is higher in infrared methods, so more light passes through the matter, which is good, but with Raman we do have a smaller interaction depth, but we also have the Raman scattering effect. And basically through that we are able to detect up until 1 to 0.1 mass fraction. So it's highly suitable for search for organics on surfaces where the location might not be readily apparent from the color or surface texture alone. So we are able to identify and characterize the matter or the microfossils in the matter at much lower concentration. So this is really useful for Mars or any kind of sample where we want to find microfossils embedded in sediments. Now I will just quickly explain you guys what spectroscopy is, because some of you might not know. I didn't know either before reading the paper. So basically in spectroscopy, incoming light hits a matter, and there is an energy exchange between the matter and the light. And with that, protons are exchanged, and this can be used to characterize the matter. The light is shown through a prism, which refracts the white light and splits it into colors, which gives us the different wavelengths. And wavelength we can think of as being a frequency of a light wave and the distance the wave can travel. We have longer wavelengths, which are lower frequency lengths, but they can travel longer distances. They are usually in the colors red, orange, yellow, green. And we have shorter wavelengths, which are at a higher frequency of light, but can travel shorter distance. They are usually in the colors blue, indigo, and violet. This colorful light points in different directions, and we as viewers see the colors in a single band as absorption lines. An absorption line is basically dark lines on a spectrum, which, depending on the location where the absorption line is, we can determine which elements are found. Therefore, it is really useful to determine the element mix the matter is made of. You can see this also in the picture. Basically, from point 1 until point 3, the source is point 1 hitting point 2, the prism, which is located in a black box. So the black box kind of depicts the prism. It gets us the wavelength, and this wavelength we can see on the spectrum, which is the colorful band ranging from red to blue, and down below you see the single band with the absorption lines. Deep UV Raman spectroscopy uses light from the ultraviolet range and can therefore detect substances of lower concentrations. There are three main processes: absorption, where some wavelengths are absorbed and some are transmitted. There is fluorescence, meaning that the matter remits light and there will be a longer wavelength than the original, so the wavelength is amplified. This because the biosamples resonate, and this is really nice because this gives us a more intense or an addition of color to the incident green light of those biosamples. And third, there is a scattering effect. Raman spectroscopy belongs to a group of vibrational spectroscopies, which means that there is inelastic scatter. There's also elastic scattering, but let's just talk about the inelastic one. In inelastic scattering, there is an interaction. Basically, it's an interaction of an excitation or de-excitation of molecular vibration in the matter, in the sample. And the characteristic of this vibration determines the wavelength of the inelastically scattered light. We can measure the intensity distribution, and from this we can deduce information about the vibrational structure of this illuminated substance. This you will see is important because with the Raman spectroscopy we basically get two bands. I will talk about this in the results, a D band and a Chi band, and this Chi band is a result from this scattering effect. But let's talk about that later. On the picture you again see the range where the deep UV Raman spectroscopy operates, and down below you also see the different wavelengths used in Raman spectrometers. So, main study goals. There were three main goals. Firstly, they wanted to characterize the Raman spectra of kerogenous microfossils, so of organic matter, using deep UV laser excitation wavelengths, and compare these to spectra by visible wavelengths for similar microfossils. So compare them to IR methods, for example. Secondly, they wanted to explore changes in features of deep UV Raman spectra with increasing thermal maturity of the matter. And thirdly, to inform Sherlock science by assessing various metrics used to determine the relative thermal alteration of macromolecular kerogen-like structures to better understand the potential preservation of relevant biosignatures. So the third one is basically only about informing Sherlock which samples are of high priority and which are not. For the study, they used fine-grained silicified stromatolite samples, which can be found in shallow marine environments. You can see how they look on the picture. There was only minor thermal alteration in the samples, so the highest altered was at temperatures 200 to 450 degrees Celsius, and they were all analyzed with deep UV Raman spectroscopy. For the results. So let's talk again about those two main bands of kerogen. They always show up in a sample which contains kerogen. Kerogen is made of carbon, hydrogen, oxygen, nitrogen, sulfur, whereas carbon is usually one of the main compounds. Here, those two main bands are a disordered and a graphitic one. The disordered and the graphitic one, they're both found at different layers in the sedimentary rocks. And in short, if you detect a band of kerogen, then it's for certain that you have a molecular biomarker. But why are the different levels important? So there is a thing called thermal maturity. It means that the matter, the composition of the matter can change due to pressure, heat, and time. In the study, they checked the peak center position of the bands, the intensity of them, and the variation among different microfossils, and found that they correspond to a function of thermal maturity. So from the thermal maturity, they can also estimate the kerogen maturity. And they found that in low maturity, they have a broad D-band. We've increased the D-band transitions into a G-band, and with high maturity, the D-band is usually absent and only a G-band can be found. So from that, we can see that from the different levels where the G and D band is present, we also know basically how old the kerogen is. And therefore we can pinpoint a timeline to the sample, which is really useful if we want to know exactly when this organic matter may have existed as a living object or living organism. And the G band is only detectable because we have those Raman scattering effects, which kind of resonate much more than we have with a typical IR infrared method. But as a takeout from all of this, because there's a lot, basically if there is a sample found by Sherlock which depicts any of those bands, a D or a G band or both, we know that this sample is kerogen rich, and it should be prioritized if we want to sample it or tube it, collect it. It is a high priority sample for sure. The microfossils found in the study were mostly section embedded microfossils, either benthic or planktonic photoautotrophs or prokaryotic microorganisms augmented by eukaryotic architarchs. I've put what it is exactly at the bottom of the slide, so just take a look and stop the presentation if you want to read in detail what those what is a benthic photoautroph, for example, or what is eukaryotic again. So just for the recap purpose. Carrying on, here you can see a picture from the study. For explanation, let's just go through it real quick. So on the top, for example, we have River Wakefield. So we have a sample from River Wakefield, and you can see in red the box where they detected kerogen bands, and then on the colorful picture you see the band of kerogen in green. So the biomatter resonated. So this is called the fluorescence in Raman spectroscopy, if you remember from the previous slide. It re-emits this light, and we see it as a green band. And we see the stuff where there is not much kerogen inside as blue. So the wavelength is blue, and this is usually the rock. So there are just 3 different samples where you can understand a bit better what the result exactly was. As a summary of the paper, it tried to find, is kerogen a possible evidence for past life? And we can conclude, as it found microfossils, which we can which we can pinpoint back to Precambrian fossil records, that Carrigan is indeed a possible evidence for past life. We know now that deep UV Raman spectroscopy can characterize geochemical samples and therefore is useful for Martian sample collecting. We also know that the thermal maturity in fossilized organic matter can be seen as a function pointing directly to the maturity of Carrigan. We can use the early rock record as a point of reference for a timeline, but now we have expanded it with the record of Carrigan. Therefore, it is really useful as well because we know that Mars has lacked plate tectonics for most or all its history, and therefore ancient sedimentary rocks are likely to have experienced lower thermal alteration compared with rocks on Earth. And these factors support as well that Carrigan is really important for the Perseverance rover, and return of those samples seem to be promising for more rigorous analysis on when they return back to Earth, that we can analyze them much more. And this helps in the end with strategies where samples are collected for life detection. So we know now which samples are of higher priority and which ones we can discard. As a personal opinion, I think this study is really important and for sure helps us in answering some of the big questions, or if life somewhere else in the universe exists and what the future of life is on Earth and beyond. They aim to support Sherlock with the record of Carrigan, and I think they achieved that. But the study is a bit more diagnostic than Sherlock, as it used a 1 micrometer laser spot, whereas Sherlock uses a 50 to 100 micrometer laser spot, meaning that the spatial resolution was much higher in this study than with Sherlock. So Sherlock is less diagnostic. Therefore, I think that it's really unlikely that the analysis performed during the Mars 2020 mission will be capable of resolving any individual Carriganous microfossils, if even present, and detect them in situ on Mars, unless they are much, much larger than typical Precambrian fossils on Earth. So therefore, Sherlock can detect them and think, oh, okay, this has a Carrigan-rich compound, and can sample it and then send it back to Earth. But we will not know for sure until the samples are sent back. And this also leads me to the future outlook. So Sherlock detected a few days ago, actually, 2 distinct periods when the Jezero crater had presence of water. They don't have an exact timeline yet, but I think it's around 3.8 billion years ago. And then more than a billion years later, the crater contained a saltier brine producing perchlorates. Therefore, the Sherlock found 2 liquids from different rocks, from different ages, with different chemistry, and it also was able to detect organic molecules, confirming that habitable conditions once existed in Jezero crater. So we have once more much more evidence that there was at one point some life on Mars. And therefore, as a future outlook, because this news is really new, I think I found it like 4 or 2 days ago that it was posted. I think it would be really cool if from those collected rover data we can now develop possible strategies on how we could generate or maybe even regenerate life on Mars. And further, the Mars rover detected priority samples. So it will be really nice to see how NASA is going on that mission to collect those samples, to tube them and send them back to Earth. I think this will be a really complicated mission to do, but might be really interesting when the samples really arrive on Earth and we can study them rigorously to really find out what kind of microfossils might have existed on Mars once upon a time. So I think the future outlook is quite positive, and I'm really happy about the paper that kind of gave a good back structure for Sherlock to do his job and his mission on Mars, so that it was kind of successful in finding some samples which are on the high priority list to be analyzed further. Yeah, but with that, I think we're at the end. Here are my references. I don't know if you noticed, but I put all the references in the header of the slide, so as little numbers. So if you're interested in anything, you can check it out here, and you have the direct links. If you have any questions, just post them down below. And thank you very much for watching.